I leave you the stage. The stage is yours, Dmitry.
Okay, thank you, Professor Gaudreau. Thank you for inviting me to do this seminar. And yeah, it's a kind of a recapitulation of the last beginning of this year seminar. And here we will talk about, in general, genome engineering, particularly introducing the CRISPR and different aspects of the engineering which CRISPR system CRISPR allows. And particularly there will be two small sections in using the genome engineering in molecular oncology. One on very good solid paper from American lab, and then small work we did here in the same area. Well, basically, yeah, that's my talk outline. CRISPR powerful genome engineering tool, and there will be a thorough introduction to it, maybe too thorough, but Alexei told me that some of the students will benefit from the thorough introduction of CRISPR. And then we will talk about application of CRISPR in colorectal cancer modeling. And the last part will be on the engineering tyrosine kinase inhibitor resistant cell line, which is relevant to non-small cell lung cancer work done here by us.
So if you allow me to interrupt, I just would like to say once again, this seminar is for you students. So if you have questions, Dimitri
Yes.
is okay if you interrupt him at any time. He will go into the very specifics of the systems you are manipulating. So this is the right moment. If you have a question, please ask. This seminar is for you.
Yes, yes, of course. Yeah, you could interrupt me, and maybe I will be not specific enough. Please use ask questions at any time. Interrupt me. All right. So even before we go into details, the most important thing which should be in your head after the seminar that CRISPR is a powerful artificial programmable nuclease method. It's powerful because it's simple and in the sense that it allows easy design for targeted modification. Normally, only two key components are needed, sometimes three. And it's so simple that even so-called do-it-yourself CRISPR culture, kind of garage genome engineering appear, at least in the United States. And a kind of basic science use, generally in genome engineering, CRISPR in particular, is a kind of reverse genetic. You modify your gene, you look at the phenotype. Very relevant to oncogenesis process. Mutation in the gene leads to cancerogenesis. It's as well unique in a sense that unlike other methods, which we'll consider in a second, it allows whole genome manipulation. Whole genome manipulation for knockouts, for upregulation and downregulation. In that case, it's called CRISPR-A and CRISPR-I. But the reason for it, because basically you could build a library of the targeted agents, CRISPR RNAs, in order to achieve it. And here you won't be only asking what this particular gene do for your phenotype, or knockout of this gene, upregulation or downregulation, but you could ask the question, what the set of the genes which will affect phenotype by doing the whole genome manipulation with a CRISPR library. So kind of a jumping out of basic science, it's already in use in crops and animal engineering. The plants and animal with the new features developed by genome engineering, particularly by CRISPR. And there is a still high expectation for use of CRISPR in gene therapy. It's so versatile that many alternative approaches of using CRISPR develop. One of them was mentioned as high sensitivity DNA RNA detection, in pathogen detection, and in this COVID epidemics, the CRISPR method has been employed, develop alternative to a real-time PCR for detection of the SARS-2. kind of important note. Maybe a few of you seen those papers. So yeah, before the CRISPR came on the board, there was a number of alternative methods for genome engineering. First was meganucleases, then the Zinfinger nuclease came, and finally TALEN. Those are all belong to a concrete set of protein DNA binding type of engineering, where nuclease domains, DNA independent nuclease domain, brought into target by either combination of Zinfinger binding motifs or the combination of TALEN binding motif. TALEN stays for transcript activator-like effectors, and it's proteins discovered in the plant bacteria, plant pathogens. The uniqueness that their domains is basically specify one base pair, very rare occurrence. Basically, actually, maybe the unique one, where, for example, Zinfinger's a domain is specified as 3 base pair. Here's each base pair specified by a unique domain. And obviously, TALEN kind of makes the engineering easier than Zinfinger nuclease, but not as easy as what CRISPR brought to the genome engineering field. And here, of course, we have completely different targeting. It's not achieved by protein DNA binding, but achieved by forming RNA-DNA hybrid. And we will talk about it in more details later. But still, protein is involved. The whole complex is DNA, RNA, protein complex. And this type of engineering came from investigating the bacterial immunity. All of it, of course, kind of were considered in the first place because the double-stranded DNA break leads to a 1,000 increase in homologous-driven repair. The discovery made early on by Maria Jason group. And then by cutting DNA, you will be able, using homologous recombination, to make changes in DNA. And that's why those nucleases has been developed. That's why CRISPR as a nuclease itself were employed for the genome engineering. So what is a CRISPR? As I mentioned, it's a prokaryotic immune system. CRISPR stays for clustered, regularly interspaced, short palindromic repeats. Those repeats was discovered quite long ago in 1987, and later on it was discovered that they're present in many bacterial genomes. Important, there was genes, homologous genes, always associated with the CRISPR arrays, which were called Cas, CRISPR-associated genes, and they all got numbers, Cas1 till Cas12. That was discovered in 2002, and immediately the nuclease and helicase activity was predicted for those Cas proteins, and it was suggested that they may be involved in DNA repair. But later, it's important to emphasize that between those repeats, basically inverted repeats, forming some structures potentially in DNA or in RNA. There is a spacers, which is not related, kind of random sequences initially, but it turns out that those spacers are homologous to different phage sequences and plasmid, it was discovered. And then there was hypothesis that it is a bacterial immune system, and importantly, acquired bacterial immune system, not like a restriction immune system discovered a long time ago in bacteria. This is a not inherited immune system against certain pathogen, but the immune system which basically can be immunity which can be acquired during the invasion of interaction with a new pathogen. Well, this hypothesis was confirmed in Streptococcus thermophilus in 2007, but it took a long time to really figure out the mechanism of action for this immunity, which was, of course, done by the group by the labs of Jennifer Doudna and Emmanuel Carpentier in 2012. And of course, you all know that this year they got the Nobel Prize for this discovery. Immediately, because it was a nuclease, it was realized that you could use this mechanism for genome engineering everywhere, in human, in all other organisms. And there was first paper in 2013 by Fan Zhang group, which was first to show that you could use this platform in engineering human cells. So, but how it works itself, without considering how CRISPR array is acquired, after it acquired, after meeting certain pathogen, after a new invasion, the pre-CRISPR RNA get transcribed. Along this transcription, the parallel gene is transcribed called trace RNA. The pre-CRISPR RNA get processed on individual CRISPR RNA, and then complexes form between trace RNA and the CRISPR. And after that, if the perfect match in a pathogen is found, and this is found by work of Cas9 invading this complex of RNA into present foreign DNA. If it's perfect match, then the nuclease is activated and this pathogen DNA get cut. Importantly, next to the CRISPR sequence, there is always a so-called PAM element present. We will talk about it in a second. So you could do a double-stranded break, and analogous to a previous genome engineering platform, you could use the system for genome engineering. Any questions here? Maybe I wasn't clear at some point. Please ask questions at any time. And then, of course, if we look a little bit into more details and talk about the language around the CRISPR platform, the Cas9, that's a protein nuclease, which involved in interaction with a CRISPR RNA DNA complex. And the complex itself, CRISPR RNA and Cas9, is double-stranded DNA cutting enzyme. You could make a trick, and instead of transcribing trace RNA alongside with the CRISPR, you can basically make one transcription frame and just link the trace RNA with a CRISPR, and that way you will create active still RNA, which is called single-guided RNA. Single-guide RNA. And this is a trick practically universally used these days, although some researchers still prefer to express them separately. Then basically, I mentioned the PAM. This is a PAM stays for protospacer adjacent motif. And basically, there is no this motif present in CRISPR array itself. It's only present in a foreign DNA next to a protospacer as a opposite to a spacer in terms of the sequence. And only the invasion and cutting will occur, only PAM is present next to a protospacer. And PAM basically is important for the action of unwinding DNA. If this motif is not present, DNA won't be unwinded at any place, but specifically where PAM is present. And then Cas9 will try to insert the CRISPR RNA associated with it. And if there is a perfect match, the complex, stable complex formed, there is a protein transition occur, which brings the nuclease domains, separate nuclease domain for each strand in the right conformation, and then cuts occur. A few details, as gRNA is about 20 base pair in length. Normally it will be unique in a human genome, but because the potentially some mismatches could be tolerated, particularly further from PAM, then there's still potential for off-target action, and this is always in the minds of the researchers to show that whatever you're doing, you don't induce too much off-targets. For that specific sgRNA develop with a low of target action. And then the cutting is blunt, although it is kind of questionable, maybe 1 base pair overhands. So basically a double-stranded cut occurs with blunt ends. Worth mentioning here that there is an alternative Cas9 platform. This is type 2 CRISPR system. There is type 5 CRISPR system, which has a different CRISPR RNA arrangement with 23 base pair targeting and very different PAM requirements, where it's T-rich, TTTV essentially. Obviously, those platforms could be used. An AT-rich platform, you will use Cas12a, another one, otherwise called CPF1, and on normal sequence you will use the Cas9. Both platforms use, and Cas9, of course, much more often, more versatile. If we talk about the protein composition of Cas9 itself, the main composition of Cas9 itself, again, very important domain, the domain, PAM interaction domain. This is involved in the PAM recognition. We will look at 3-dimensional representation of the structure in a second. And the REC domains are homologous to basically to the topoisomerases, right? And they're involved in unwinding the DNA next to the PAM, and then invasion of CRISPR RNA happens. And then RUF-C and NH domains are nucleases. Importantly, those nucleases or single-strand DNA nucleases, active sites are in different places and in different domains, and you could separately inactivate it, making a nickase, single-stranded DNA cutting enzyme, out of double-stranded DNA cutting enzyme. And you could cut either one or another strand. And if you inactivate both of them, you will create so-called dead Cas9 and convert it, in this case, into sgRNA-guided DNA binding protein, making it basically analogous to other DNA binding protein, but with completely different mode of recognition. This is as well gets used a lot in using CRISPR system for gene activation or gene repression by attaching repressors or activators, different effectors. Again, as I mentioned, we will look at the kind of representation of 3-dimensional structure. And this reminds a little bit of polymerase sitting on a double-stranded DNA. Here's kind of RNA emerging out of it, if we talk about transcription. But here, of course, is completely different structure. It's a DNA-RNA hybrid. And what emerges out of this structure and kind of become in solution is a second strand, so-called non-target strand. This strand called target strand, it binds the RNA, CRISPR RNA. So it's target strand, and this is non-target strand. And it's important to mention it because some of the methods developed recently rely on the presence of this non-target strand outside of the double-stranded hybrid in solution, making the base pairs of the strand available to enzymatic action. And that's basically a mechanism of it, because this second strand is not within the protein structure, it's outside, and at least part of it available for manipulation, for enzymatic manipulation. Importantly, you made the double-stranded break, but again, that's not the end of it. Of course, the actual engineering happens with the use cellular DNA repair system, right? And it's important to know that there is a major DNA repair system present in all cells. It's a non-homologous end joining mechanism, kind of much more active, and homologous driven recombination, homologous recombination mechanism, which usually normally would use the second chromosome to repair a double-stranded break. A double-stranded break is the most lethal damage you could have to genome. If it's not repaired, basically most of the time cell will die and will go to apoptosis. Cell will try to repair immediate double-stranded break using one pathway or another pathway. And of course here, when non-homologous end joining get involved, then you will basically insert, make small insertion or make small deletion and basically disrupt the frame if it's coded gene. And in that way, you could disrupt the gene, make the knockouts. If it's homologously driven repair, then here, if you instead of will trick your cells, instead of using the homologous chromosome, will provide some double-stranded DNA or single-stranded DNA, then those can be used in homologous recombination as a donor DNA. And in that case, you could make a substitution or make controllable insertion, a deletion. And this is, of course, much less prominent mechanism. We'll talk about fight between one and another in a second. But as I mentioned, worth mentioning here that the other function, other potential application is activation and repression. If you use dead Cas9, attach either activators or repressors, then you could use same as when normally TAL or Zenfingus would be used. In that case, you could use a CRISPR platform for activation and repression. And for that matter, unlike other platform, you could use for the global gene activation and repression because of the possibility of designing the whole genome library of CRISPR. Okay, so moving further, yes, during genome engineering, when you engineer, usually it's random outcome. And non-homologous end joining using UQ proteins and combination of different kinases and association of ligase will try immediately to capture the end, and after small processing of the ends, will ligate it, and in that way you could, disrupt the gene, knock out the gene just because of frame shift. There will be premature stop codons soon. That is quite efficient way, and that very often how the CRISPR system with double-stranded break is used. But then the second repair mechanism is homology-driven repair. We already discussed it, but important here that it's used by the cells only on a part of the cellular cycle, and then still on the background of available non-homologous end joining. This is, of course, required and quite often needed for precise editing, but it will be always kind of marred by the presence of non-homologous end joining. And to find your precise editing, the one you require, you would have to shift through the number of clones where non-homologous end joining did you some damage. All right, precisely because of that, because people didn't want it to fight this damage, there was a successful attempt to develop a different approaches to genome engineering still using the CRISPR, which called base editing. And here it is important to remember, as I mentioned initially, that there is a non-target strand is available to solution and available to enzymatic action. There is enzymes available in nature, which is a cytidine deaminase, and there is as well the one developed specifically for this approach, adenosine deaminase, DNA adenosine deaminase. And in this case, if deamination of cytidine will happen, it will become uridine, and eventually it will be converted to thymine. The same here, adenine will be converted to inosine, and eventually after a few cycles, it will become a guanine. So C to T and A to G substitution, which call transitions, are available without damaging a non-homologous end joining, because here you could use either dead protein or nicase protein, which will not induce non-homologous end joining. And in that case, for those substitution at least, you will do a precise engineering without mutagenic effect of double-stranded break. Importantly, if that happens in non-coding C to T substitution, then on coding it will be G to A substitution. Similarly here, if we're making the changes on coding strand A to G, we will convert T to C. So all those four transitions are available for genome engineering on your gene. Of course, for that, it would require the presence of PAM within the certain area. And importantly as well, worth mentioning here, because CAA could be converted to TAA, to a stop codon, CAG to TAG, CGA to TGA. So you could use the Q or arginine to basically make a specific stop instead of making the knockouts using cytidine base editor rather than using double-stranded breaks. That gets used currently in the CRISPR genome engineering. So I forgot to mention that this approach has been developed by laboratory of David Liu at Harvard Medical Schools, and I think at some point they probably will deserve the separate Nobel Prize for the development of this technique. I already kind of a little bit mentioned it. It's not always you will be able to use this approach because particularly for base editing, because the window where the non-targeted strand, part of non-targeted strand available to solution is pretty small, and on a particular distance, 15 base pair from a palm. So you have to be lucky. So your palm have to be within 15 base pair from area of modification, and three to five bases here will be available for the base editing. This is not unique to base editing, although window here is smaller. In case of HDR as well, your palm cannot be too far from the place where you make a substitution using the homology-driven repair. Basically, it has to be 10 base pair one way or 10 base pair another way. So basically, a 20 base pair window here. If there's no palm within that, your modification probably will be not very efficient. You will have to shift through hundreds of clones to find what you require. And I won't be mentioning too much about this, but using different type of cytidine deaminases, you move in this window a little bit, expanding the possibility of targeting. Precisely because of it, maybe there is a alternative, the completely new CRISPR 3.0 method has been developed just last year, which called prime editor. And it is basically now chimera of Cas9, not with the deaminases, but attention, reverse transcriptase. It's a chimera of Cas9 with a Cas9 nickase with a reverse transcriptase. And how you will be able to use it? Well, you have to design a special so-called prime editing guide RNA, PEG RNA for short, where the 3' end of this normally interrupted here, CRISPR RNA extended, and precise design of this will allow you to use reverse transcriptase to make any type of substitution you want. And how it works? Basically, as this non-coding strand is available in solution, by extending your CRISPR RNA, making it PEG RNA, you potentially can form this second DNA RNA hybrid with a non-target RNA if your most 3' contains perfect match with a nicked, importantly nicked non-target strand where Cas9 binds, Cas9 nickase bind. Nickase will nick. It now can form a complex, and of course it's a RNA DNA complex, and this has 3' end present, and this is basically a primer for the reverse transcriptase action, right? So if you attach reverse transcriptase, bring the reverse transcriptase to that site through the fusion with Cas9, then you can potentially extend from this primer DNA further, right? And if you introduce certain substitution, now you making the editing, DNA editing here, kind of a little bit more here in terms of how it happens after extension. This so-called flap appears, and of course it could go one way, preserve the wild type, or can go other way, and so-called base excision repair machine gets involved here. You can use certain tricks to enhance preservation of your addition here, making a NICs additionally, but we will not talk about it too much. Important just to understand CRISPR 3.0. It's a modified CRISPR RNA called PEG RNA, and then it's a fusion of Cas9 with a reverse transcriptase. It seems like very powerful techniques. Now you not rely only on the transition. You can make transversion, for example, from pyrimidine to purine, or from purine to pyrimidine, just because you could encode it in your PEG RNA. And then the second important point, that now you're not as restricted in terms of the pump position, because you can move those much further. It's not a fixed window. You could extend from this primer further on, make a longer PEG RNA tail, and then you could make addition somewhere here, and so your pump can be further away than when you're using non-mutagenic base addition, right? So at this point, any questions, particularly concerning the prime editing? All right, hopefully.
Have you used, Dmitry, this technique?
No, I didn't. We actually got all the reagents, and I was prepared to go and use it. We've been successful making this genome engineering by using standard technique, and so I stopped. And then I heard kind of from other colleagues that they haven't been successful in using this technique yet. And then there was one paper from a group where they compared it with a base editor developed by them, particularly with they compared their prime editing with base editor with prime editing, and then they had shown that it seems like active to some extent in HEC293s, but very little activity in other cell lines. So it's still kind of up in the air. It's very promising technique, but it's still up in the air how effective it will be. Maybe it remains to be developed further, as it was with the base editors from a David Liu group. By the way, this technique as well developed by David Liu group. Very inventive, very inventive group of very inventive lab in terms of the developing new genome engineering tool. All right, more questions?
I have a question.
Huh?
I have a question, sorry.
Okay.
You talked a lot about CRISPR-2 and CRISPR-3, but how did they manage to design it? Was it already, could they find it in in a bacteria, or they design it from the bottom?
You mean designing the sgRNA for targeting specific gene?
Yes, for example, how they do combine RT, the reverse transcriptase and the Cas9, how they do this?
Well, basically, in order, they took the bacterial reverse transcriptase, not bacterial, I'm sorry, MLU, the viral reverse transcriptase. They actually initially used the wild type reverse transcriptase. The activity was kind of low. They did the genome engineering on it, finding a certain nearly, if I remember correctly, 5 mutations, which seems enhanced its activity in mammalian cells. And yeah, it's a bacterial reverse transcriptase, just genome engineered further to make it more efficient.
If you allow me, Dmitry, perhaps I will answer this question because I know what they know and I know what they don't. So basically, you can fuse any gene with any gene and you make a fusion protein. So you just take the gene of reverse transcriptase, you remove the stop codon, and you put in the right frame whatever you want afterwards. So this is what researchers did here. And so the initiating methionine of Cas9 will be just an internal methionine, but you will make a single protein having the two functions. It's called a fusion protein.
Fusion protein, precisely. Yeah.
So you can design whatever you want. It's bricolage, it's DIY.
That's right. Yeah, you could attach different effectors, activator here, and it will activate if it's in promoter, or repressor, and it will press if it's in promoter. Same way, the same fusion was used when the base editors has been developed. It's simply attaching the deaminase, either CT deaminase or AG deaminase, to the Cas9. Cas9 here plays mainly the role of bringing this enzyme, reverse transcriptase or deaminase, at the right place to make it close to the basis it will deaminate, or to the basis from which it extends, to the primer from which it extends. So Cas9 here is basically a carryover of the important enzymatic function. So that's how it works. All right.
Thank you.
Okay. And yeah, kind of finishing this section. So we kind of came through the number of different tools. Cas9 is the most widely used. It has NGG as a PAM, but there is different natural orthologs, different sizes, which important, and you could insert it probably in the viruses for viral delivery, different PAM specificities, which could important. You could engineer Cas9 to change the PAM. This is precisely important because then you could find the PAM at the right position for this particular window, right? Either for HDR or for base editing. And then importantly, a rational design of Cas9 has allowed to develop so-called NG PAM, Cas9 NG, where instead of NGG, the PAM become NG, obviously very dense targeting. And it's really widely used now. I mentioned about different types of the Cas9, Cas system, Cas12a, with a completely different PAM, probably will be used more in AT-rich sequences. You can make Cas9 nickase. Specifically, you will use it for the gene therapeutic programs, for example, where you don't want to introduce additional mutations by the activation non-homologous end joining when you using the double-stranded break, when you use a nuclease. And then, yeah, we basically went through it. Cas9 base editors. This is a deaminases attached to Cas9. And in case of prime editing, this is the Cas9 reverse transcriptase chimera. And it remains to be seen if this technique will take off. But at least it's a really fantastic idea from the David Liu lab. All right, so we went through this through the genome engineering toolbox. We've considered practically all type of application, and always still important to remember that by doing all those modification, even when you do the base editing, you don't do the C to T edition, right? You're doing C to U. So in all cases, the internal DNA repair machinery involved in non-homologous end joining, non-homologous recombination, in the base editing, in the prime editing, because the base excision repair machinery will get involved to convert those flips. All right, that's kind of with that last note. We will move to the second part. We will show the example of using genome engineering, particularly for the cancer in cancer. And I should emphasize that the genome engineering considered as a gene therapy in cancer. You could imagine that if you convert mutagenic, mutations, cancerogenic mutation back to a wild type, you could use a genome engineering as a gene therapy tool. But of course, it's important to deliver to each cancerogenic cells, and this is the biggest problem. And as well, there's usually multiple genes involved, so you have to do the therapy, do the genome engineering, gene editing on all of them in order to treat. But the other part in using the genome engineering in molecular oncology, it making cancer cell models, for example, for high throughput drug screening, and that is widely used and basically available for the use in molecular oncology. And the rationale for the use of genome engineering cancer research is simple. Cancer is sequential accumulation of mutation leading to malignancy. There is a certain driver, so-called driver genes through the driver pathways involved, and the subset or set of driver genes will lead to a particular type of malignancy. Then, of course, CRISPR system is ideal to do the targeted genomic modification, one which emerged in those driver gene, and it's a perfect mechanism for acquisition of cellular models for a specific cancer type. And always important to remember, as I mentioned, even for the cancer treatment, right? You're not gonna gene engineer correctly all cells in the pool. There's always cloning processes involved, and then either through a dilution cloning or through the use of certain drugs, for example, pyramicin, you will select from a pool of cells where it's combination of wild type, other mutation and your required mutation. You will isolate the clone carrying the required modification on a subset of gene, on your particular gene, or on a subset of gene, and that is always involved when you are making the cellular models. The paper I'm going to discuss is on colorectal cancer. And colorectal cancer, just briefly, is the third most common cancer in men, second most common in women. There is more than 1 million cases worldwide and a lot of deaths. It's characterized particularly by chromosomal instability and microsatellite instability, and those two are involved, present at the colorectal cancer. And the cancer itself starts from transforming normal epithelium to adenoma, and then proceeding to carcinoma, and ultimately becoming invasive metastatic tumor, a very deadly one. And quite a long time ago, Ferran and Vogelstein elucidated specific pathway essential to development of colorectal cancer and suggested a model consisting of accumulation of mutation in multiple genes, and all those genes regulate cell growth and differentiation. And in a relatively recent paper, sequential cancer mutation in cultured human intestinal cell line, these two groups of Gerd Kops and Hans Clevers had used CRISPR to engineer a set of genes, which we're going to talk about it, which one in a sec, in order to try to model the colorectal cancer using the genome engineering. First and important that the reason they've been able to do it, because they mastered the stem cell colon culture, right? So this is the crypt in colon, which is normally its integrity maintained by the presence of stem cells at the bottom of the crypt. Normally it will, change the damaged cells differentiating through different parts of the crypt. But then you could collect those, and at the presence of a certain growth factors, Wnt, spondin, epidermal growth factor, and noggin, you could maintain those practically indefinitely in culture with stem cells. And important, those signaling factors used in maintenance of this culture play important role in controlling the growth of normal epithelium, right? And in this Ferran-Vogelstein model, they suggested that there is a sequential mutation occur in the 3 tumor suppressor APC, adenomatous polypus coli, P53, and SMAD4, and then specific KRAS mutation converted it to an oncogene. And then sequentially, APC got knocked out, then it became early adenoma, then KRAS mutation occur, it's intermediate adenoma, then SMAD4 loss, this transcription factor in TGF-beta signaling pathway will emerge late adenoma, quite already dangerous situation. But then knockout of P53, the tumor suppressors involved in majority of the cancers, will lead to emergence of carcinoma. That there was their model. They're important to say that those growth factor is involved in signaling, in the Wnt signaling through APC, in a TGF-beta signaling. In case of loss of APC or loss of SMAD4, you basically will lose the requirements for having those for cellular proliferation. And then you could use withdrawal of those factors for selection. You make a knockout of the cell, and then you select a specific pool of cells will be preferentially grown in the absence of Wnt and spondin, then having mutation APC. Withdrawal of noggin will lead to the preferential growth without the presence of wild type SMAD4. And in case of P53, they use Nutlin. This is a non-drug to favor the growth cells with mutated P53 protein. And basically that was their idea. They wanted to model the appearance of this cancer and look if it's indeed how the colorectal cancer tumor look in human. And for that, they use a CRISPR. And essentially, to do that, each time you will generate a number of sgRNA for targeting. Here they use exon 15, generated 4 different sgRNA. For 2 of them, at least, they had shown that they've been successful in disrupting both genes on both chromosomes with a different, obviously, mutation on each allele. And importantly, they had used and had shown that it can use 2 different sgRNA. This is sometimes important to generate knockout mutants using different tools, different targeting, because as I mentioned, there could be off-target effects, and you could ensure that the effect, phenotypic effect you will see is not due to some off-target effects you have in your clone, right? Ensuring that with 2 different tools you see in the same phenotypic effects, you're quite sure that effects are due to the knockout of APC and not to some potential off-target effects. Basically, after that, they generated the similar tools for each of their 3 tumor suppressor gene for the APC, P53, and SMAD4. But more kind of complex task for that was generating glycine 12 to aspartic acid substitution in KRAS, and that they had used then homology-driven repair process here. They made a cut, developed sgRNA, making a cut close to the coding part of G12, and then used oligonucleotide carrying aspartic acid codon here and basically making the homology-driven repair, selecting clones. They've been able to select clone where one allele was knocked out and another carried precisely the G12 to D mutation. And on top of it, they generated the way I had shown for APC, withdrawing the hormones for the required for the maintenance through the APC. They generated a knockout on APC, on top of it P53, and then on top of it SMAD4. In each case, using its own combination of withdrawn or added factor for selection. So this kind of selection, not using the puromycin, which you usually use natural factors by withdrawing and adding specific factors. And here are the Westerns showing the knockouts on P53, SMAD4, on combination of knockout. And here at the end, the APC knockouts had shown in Western too. So basically, they generated either mutation in KRAS, on top of it, knockout on APC, APC plus P53, and APC, P53, and SMAD4, triple knockout, and then mutation in KRAS. Right, so they have this all different combination of cellular clones. Importantly, actually, after the KRAS mutation, they have seen the change in the organoids, which reminded more adenoma rather than kind of more flat epithelial colon cells. By characterizing all those different mutants, and particularly the final one, which leads to a malignant invasive colorectal cells, they say that KRAS G12 combination, having all 4 mutants organoids, can grow in the absence of all stem cell niche factors, which needed to maintain the stem cells in vitro. And only quadruple mutants grows as invasive carcinomas in mice xenotransplantation experiments. Although overall proliferative activity of triple and quadruple mutants is similar, only all four mutations, 3 knockouts, and substitution will lead to basically to a carcinoma. And then they had shown that indeed they observe extensive aneuploidy when APC and P53 was lost. Here is normal karyotypes, and then there is loss of chromosomes here, wrong karyotypes. And then even when all 4 mutation occurs, then there was even more loss of chromosomes, and high 75% cells was unemployed. And then, as well importantly, in their judgment, histology of quadruple mutant organoids corresponds to what the histology of carcinomas in humans provide, right? And kind of a punchline at the end, this is an example of successful CRISPR use in modeling carcinogenesis. And one can say that this work has confirmed this initial model by Vogelstein in developing the colon cancer. Any questions here? Any? Obviously, I'm not an expert in oncology, but here is a main idea: is we're developing the tools. Using those tools, we discuss how you will use those tools to make either specific substitution or knockouts. And important that sometimes you need to do knockouts with a different reagent, to be quite sure that whatever you develop is actually the phenotypic effects due to the knockout of your targeted gene. All right, and then
so students, do you have questions? Questions are for students first, but if not, I have one. But go ahead.
I think I have a question.
Uh-huh.
We're trying to do the same thing with breast cancer. But is it possible to do to use the same technique with the other type of cancers? I don't know.
Absolutely, absolutely. Yeah, the idea is the same. So if you know the driver, the driver genes or driver pathways, then basically you select this gene or combinations of the gene and do sequential knockouts of those genes, leading to emergence of those cancer. For example, for triple negative cancer, it will be BRCA1, BRCA2, and I forgot the third one. That will lead to a so-called hormone-resistant breast cancer, right? So yeah, you will basically do precisely the same thing, but you will develop the new tools, new sgRNA targeting the required genes. If you will need not only knockouts but specific substitution, making the oncogene. If oncogene is necessary for this process, then you will use homology-driven repair, or maybe, if you like, base editing, because this process is a little bit easier to achieve.
So just to answer Camille again, the beauty of this paper you presented is that they use stem cells, which are absolutely not established cell lines. We use an established cell line, which is much easier, but less physiologically relevant, perhaps.
Yes, sure.
And I would like you, Dmitry, to comment on the fact. So they were using this removal of growth factors to select the clones that were correctly edited, but do they go through a clonal selection? I mean, they just use a pool of whatever survives and grows without the growth factors, or do they still pick and characterize some clones?
You know, to my shame, I cannot answer. I read this paper thoroughly long time ago when I was preparing for it. I cannot tell you definitely. I will look and will answer you separately.
Okay. No, but I can look as well. It's a terrific paper indeed.
I absolutely, I agree. It's a terrific paper. Yeah. I don't know if it was pools or it was a specific, if it was a clone.
All right. So I let you go on, finish with your last part. And,
well, we're already a little bit over the time, but I started late. So quickly, this is not as powerful, but it's the same idea. We're talking about genome engineering in generated tyrosine kinase inhibitor resistant cell line. It will be clear in a sec what it means. And this is a kinase inhibitor resistant cell line in a model of non-small cell lung cancer. So EGFR is a molecule which is involved very often in developing non-small cell lung cancer, quite as well deadly cancer. And what happens basically, there is a particular, in this case, quite often leucine 858 arginine mutation occur, which basically make looks epithelial growth factor receptor. So the receptor which detect epithelial growth factor, right? And normally only if it's present will signal for cell to proliferate. But then with this mutation, it will signal in the absence of this epithelial growth factor, and will basically tell the cell, which should be, basically not growing, will signal to cell growth and proliferation. There is MAP kinase pathway downstream of this, and basically this mutation will look like activated receptor signaling to MAP kinase. For that, certain amount of drugs developed, they called tyrosine kinase inhibitor drugs, like gefitinib or everolitinib, and those will inhibit the signaling of that mutation. The receptor wouldn't look in the presence of those drugs as a signaling. But practically inevitably, after successful treatment with those drugs, there is a second set of mutation occurs, particularly frequent ones is tyrosine 792 methionine, which will make basically binding of those drugs to this ATP binding pocket is non-efficient, and basically essentially will make those drugs are not efficient. And obviously, it's a medical need to develop a drug which will be, effective in combination of those mutants, other mutation too, but this one is the most frequent. And that basically was our task, essentially to develop tyrosine kinase inhibitor resistant model in a lung cancer. And here we as well, not as relevant, we're using already adenocarcinoma cell line, which has a wild type EGFR receptor, but then we inserting our plan was insert those mutation and make a model at least partly relevant to developing, to screening the library and developing new drugs. And in this case, we're basically making the first substitution of leucine to arginine. Here's only homology-driven repair was available for us, so we use oligonucleotide as a donor, similar to KRAS mutation the people did in colorectal cancer. And then here we were lucky. I show you why we're lucky. So we could use cytosine to threonine conversion. So basically using the cytidine base editor, BE3 for this mutation. And basically first we're making mutation number 1. After obtaining this clone, we will make mutation number 2. And again, here I already talked about those windows. And if you guys will be doing the design, you should keep this in mind too. There's always important, you have to have your arm within the active window in order to use HDR, particularly to use base editing. This has to be in your mind. That's why increasing the changes in pump specificity for Cas9 or for CPF1 is a hot research topic, increasing your targeting. Using the USC genome browser, here we're looking at the leucine 858 position, and as you could see, those stretches as sgRNA. Coding, there's a whole bunch of them. Green should be a particularly good one. Red not as good, bad basically. Yellow is relatively good, and then dark or black is potentially creating off-target effects. So you're not going to use those. So you can pick and choose those, and basically and then compare them, right, in terms of their efficiency, because not only you want to have a cut close by, you could use this or this for that, although this one is closer, the cut occurs roughly here, but then they have to be efficient, and that is measured by so-called T7E1 assay. Previous one, in my previous talk, I immediately skipped it because a student was supposed to know. But here I will go through it. Is it okay, Alexei?
Well, they all know about it because they're experiencing it. They're using it in
excellent,
the lab.
excellent. Then I skip for the sake of time.
Yeah.
We will use T7E1 assay, and basically picking 3 of them. You could see if you used to read the T7E1 assay that C91, C92, or C94 is efficient. The C91 and C92 much more efficient than C94. So we focus on using those. And basically, if you add on top of it the donor DNA, 5 micrograms or 25 micrograms per 1 million cells, basically nothing changed in terms of any NHJ. That is the fact. But then if you actually do the NGS sequencing, you do discover that 20% of your cells will have a right substitution without non-homologous end-joining, spoiling somewhere else. But actually 74% of cells, as you could see, will have a indels modification somewhere else. Basically, those 20% pool-wide will convert to 10% double allele substitution, right substitution of T to G, basically making, instead of coding CTG leucine, making CGG arginine. You could make CGC here because it's a silent, and then you will create ASC1 site here, which could be useful if you are not able to use NGS. You could screen your colonies using the restriction enzyme, something to have in mind for you. So basically, that's it. We've been able to generate a few clones here. At least 10 different clones have been generated, and we used a couple of those to proceed further. And on top of that mutation, generated the threonine methionine at position 790. And as I mentioned here, that here we're lucky we were able to use cytidine to thymine conversion using the base editor. And here, advantage not only that this process is efficient, there won't be a double-stranded break at all here, only nicking, and obviously you have to sift through the much less amount of clones in order to select your clone, hopefully. And that was indeed the case. First of all, we've been lucky that here is a sgRNA where the cytidine is in the non-targeted DNA, because if C is here, then it complex with G, so the target is opposite, and the other strand will contain the C, precisely in solution available for modification, and on the right distance of roughly 15 base pair from PAR. And this sgRNA, if you profile it again with T701, is still efficient in terms of cutting, then efficient in binding, and obviously should be efficient in base editing. And indeed, when we look at the base edition using BE3 base editor with that sgRNA on a precise needed position, we see 40% activity on C to T substitution in this place. As well in the pool, at least 10% substitution on this different close by C, but we were lucky that this is basically a silent substitution. Of course, you can clone only with that substitution, and this one will be absent. But even if we clone the clones with both substitution, we still will have a right only at threonine to methionine substitution. And indeed, here's the sequence of the clone. Second mutation introduced on the top of the first one, where it's ATG instead of ACG, and the methionine at the position of threonine 790. So that's basically it. We've been able to generate double mutant, which will be tyrosine kinase drugs, inhibited drug resistant. And then the other group of people who use those cell lines had shown that indeed this double mutant is expressed. And then screening a library of FDA-approved, a little bit more than 1,000 compounds. They came out with the drugs which inhibit growth of those cells. And one particular one is kind of making sense because, as I mentioned, it's ATP binding pocket, and it's look like ATP, but with certain modification that could probably bind and then inhibit the cells, the proliferative signal from this double mutant EGFR containing cell line, which has been done in a laboratory at Institute of Cytology at St. Petersburg. Just quickly finishing here. So CRISPR, hopefully I convince you, CRISPR powerful genome engineering method, and you can use it, it will be advantageous to your project. It can be used to model complex carcinogenesis, particularly on the basis of presentation on colon cancer. And then importantly, CRISPR can be applied not only to introducing mutation, making genomic mutation, but to gene regulation, activation of gene or repression of gene, which sometimes is a part of carcinogenesis. And particularly dark area of oncology is epigenetic changes, which do occur, and we know they're important, but you could model those with a CRISPR as well, attaching the different effectors. For example, cytosine methylation fusion, making the CRISPR Cas9 cytosine methylation fusion, and then it will methylate targetable in directed area cytosine, which may be important for the epigenetic process in molecular oncology. You could methylate histones as well. At that point, I stop. Too much words. I just at the end I want thank you all for listening to me and acknowledge members of Intracellular Signal Laboratory at MIPT, and particularly head of the lab, Nikolai Barlev, in guiding me through the research. Stopping here, maybe now question and answer section, if you're all not too tired because of the presentation.
First of all, thank you very much, Dmitry.
Thank you. It was a little bit hectic, but I tried to do my best.
No.
Thank you. Thank you very much.
Question? No? All right.
So I should tell you, Dmitry, perhaps what they're doing in the lab. Well, in the lab, they are using CRISPR-Cas9, the regular one, to induce indels using NHEJ. And so they want biallelic knockouts. And typically they do so by transfecting the Cas9 purified proteins and the guide RNA together with a tracer. And they, just by limiting dilution, they have to screen about 100 clones in order to get a couple of
You're right. That sounds, yeah, that sounds like, a process. I usually...
Can you comment on so if we were to use the same technique in order to make homologous recombination using an oligonucleotide, would we have to screen many more, just on a single allele, but would we have to screen many more clones than 100, or with 100 do we have?
I would bet that you will. So if you get only, knockouts 2 out of 100, then the HDR will be much less efficient, and on the surface of it, unless something incredible happens, then you would have to screen more clones in order to get the specific substitution.
So something that we don't use yet is NGS. So it's quite useful, right, to analyze the population before looking for the very rare variants. The machine is available downstairs.
Right. Yeah. This is absolutely. If it's available, it's a great addition to the CRISPR technology in order to profile the pools, particularly because if you're not looking only to disruption, so to knockouts where you could use T701 to estimate, but if it's specific changes, unless there is a emergence or disappearance of restriction enzyme, as was in my case, in one case, then you have basically to use NGS to estimate the percentage of your specific correct substitution without additional non-homologous end joining damage, which does occur quite often. Many of your correct clones will be spoiled anyway by non-homologous end joining. So yeah, here with individual reads, you will see precisely a percentage of your precise modification without spoiling, modification spoiled, and indels, number of indels, and then you will be able to estimate, oh, I have at least 5% of those, so I can go ahead with 100 clones and hopefully we'll get couple of biallelic substitutions. Yes, absolutely necessary too.
So do people use ways to inactivate an HEJ in order to increase HDR?
Yes, it seems like there's a few drugs which some groups developed or advertise. I tried to use a couple of them. I haven't seen big difference, and importantly, always when you use those drugs, you only will inhibit non-homologous end joining. You will not enhance HDR that way. So but it could be helpful if they work, because less of your clones will be damaged by the parallel non-homologous end joining. So this is something to consider, but unfortunately, I could not advise to tell you use this one, because I've been successful with this one. I used a couple of times, haven't seen big difference, and basically never used it really in engineering, in a cloning.
And so the last point perhaps is when you use base edition, not only you will have to use a plasmid-based genome edition, so you use a puromycin to select transfected cells. So not only you have this selection first of cells that should be modified because they express the right thing, but also on one of your slides, you show modification in as many as 40% of cells. So it means it's sufficient to screen, I don't know, 10 clones and that's it.
Absolutely, and those without puromycin selection at all. That was just CRISPR RNA and plasmid expressing BE3. And that's it. So 40% of the pool of the cell has the required modification.
Wow! But
yes,
the plasmid is not selectable. The plasmid
the plasmid I use is not selectable. Okay. There is probably a selectable plasmid, but in my case there was no selection involved, which sometimes is advantage, right? Because puromycin selection itself could lead to a certain thing. So if one
and especially if you want to introduce multiple genetic alterations, you cannot use the tricks several times. But so it means, excuse me. So it means that for most knockouts that we want to obtain, if you don't care where you introduce a frame shift, well, quite early on of the gene structure for sure, but then you have sufficient options by base edition. This is what we should use all the time, right? Instead of an HEJ.
Theoretically, I'd have to look. I'm making this recommendation because when basically C to T deaminase appear, that was immediately suggested, and people was even saying that they developing the whole genome knockouts libraries based on the base edition knockout, introducing stop codons. I don't know how far it processed. I actually developed myself some sgRNA to use to introduce stop codons. Haven't used them, just didn't get the chance for those particular genes. I generated them, but cannot tell you how efficient it was. But it should be more efficient than, or well, not more efficient, but at least as efficient, right? But then you have a controllable stops there, not some sort of frame shift, which is somewhere you will have a stop here. Your protein will stop there. So yeah, it potentially very seems like very appealing a way to do
but then I guess the design is more complicated, and so this is why perhaps it's not as widespread as
maybe yes, yeah, absolutely. So again, here you play with the windows. And so your palm has to be at the right position and at the right strand, right? Because palm can be on one strand, another strand. It has to be at the end. Basically, your sgRNA has C in it, right? So and palm has to be within those 5 base pair. Basically, the C has to be 15 base pair from PAM, could be 14, could be 13, but not further from that. Otherwise, it basically drops immediately after those couple of moving from bases, moving from 15. So yeah, in that it's limiting. But in my search, when I looked at the number of genes in the early exons, in exon 1 or in exon 2, you usually could find the sequences which will will have a glutamine or arginine. I mainly look at glutamine because there's the 2 codons there which are available for the stop codon conversion. Then they're usually available. They're usually available at number of genes I looked.
All right. I'm sorry, I didn't mean to interrupt you.
I meant to say that in that paper, when they saying that they developing the whole genome library with stops, knockouts, they say that 97% of the genes they looked at are available to this approach.
Okay, so one last call for questions from students. Perhaps you have questions now? If not, then I will stop the session and I will keep you, Dmitry, for a short discussion afterwards.
Okay.
So students, questions or not really? Okay.
Okay, thank you everybody. Thank you
So-
for your participation. Thank you for attention. Hopefully it was useful for you.
All right. So bye-bye. Thank you again, Dmitry. And then talk to you later.
Thank
Bye-bye.
you very much.
Thank you. Thank you, Alice.
Goodbye.
Good night.